syllabi and other things already has been given. While like after exam, we need to continue our classes to complete the whole course. So in today's session also, we'll try to cover at least one family of one. So as part of the syllabi given in your course, family. This Paramyxoviridae, one of the disease, the Newcastle disease, which already Professor Rajakumar has taken in the last day. Now, like read that infants, and then unfortunately, you could not share the slide at the time. Hopefully, all of you have go through it. It's quite an important disease for us, and I think you can consider it as a review of the chapter. Beside this, we are going to discuss another, in fact, here shortly Rinderpest and the distemper. We need to cover it in today's session. In the previous two classes, we talked about the double-stranded virus, RNA, the infectious versus. And the previous class, we talked about the Rio variety, where is RB and plus infection. So the disease like the blue tongue, then African horse sickness, and the rotavirus diarrhea that we have discussed it. I was expecting that you'll be asking some questions on that. Please do ask your queries. Now everything is there so that we can address and we can. Now I'm just sharing my slide. Can you see my slide even in the mode? Yeah, can you see it? So this is another RNA virus family of Paramyxoviridae. This particular grouping is really based on the type of infection they produce. And this is like this group of viruses, other than the mycelium, they target mycelium, just adjacent to the membrane. For example, the lymphocytes in the patches, the virus infections will be there, or in the subepithelial tissues causes inflammatory changes. That's why this group of virus are designated as Paramyxoviridae. Para means side by, and means mucous membrane. In contrary, we are having another ortho mean true, and that truly they infect the mucous membrane. That include the influenza viruses will be discussed. So this Paramyxoviridae family is a collection of a large number of viruses. This group of viruses categorized in order Mononigavirales. The whole name of the word mono means single, having single, they're non-cyclus. So in the previous what we discussed it, Rio as well as the BioRNA, the example of segmented virus, double-stranded virus. So here they are nega means the negative sense. These are the collection of virus, negative sense RNA virus. When we say the sense your mind, so this virus must require their specific enzyme. RNA-dependent RNA polymerase, RD, we say, okay, RNA-dependent RNA polymerase enzyme. Very good. So that enzyme, the virus never get it inside the host because the incorrect enzyme. We don't have that enzyme in the animals, or so. Where from the virus, they must carry that enzyme in their nucleocapsid structure, and the virus genome must be the to the enzyme, they replicate inside a host cell. And towards the assembly stage, they will pick up some of the molecules for the next replication. So mononega virales. Virales means an order. Suffix virales means it's an order. So this group contains 9 different families, and there are many important infections in animals as well as in the human, including poultry. So we are not going to pick up all these infections. Today we'll be talking about this Paramyxoviridae family. This is the Paramyxoviridae family. So some of the closely related mononega virales are the Rhabdoviridae, Filoviridae, Borna, and the pneumoviruses that we'll be covering in the subsequent classes. Okay, so our again concise to our concern about the important diseases. So this Paramyxoviridae, the subfamily Orthoparamyxoviridae, Rubella. This rubella virus that mainly causes mumps in human. All of you have heard about. Probably some of you might have suffered during your childhood the mumps infection, swelling of these glands. So this rubella virus is. Then Evula virinae is another subfamily. Virinae, V-I-R-I-N-A-E, that indicates this is a subfamily. This Evula includes an important poultry disease called Newcastle disease, NDV virus infection. And in the Orthoparamyxoviridae subfamily, we are having the morbillivirus. This morbillivirus includes this canine distemper virus, Rinderpest, and PPR, small lineal morbillivirus infection. Out of this, like the Rinderpest has already been eradicated globally, and this is a great success of the veterinarians that we could able to eradicate it from this earth. The Rinderpest infections. And earlier class I told you, this is one such a viral infection which gave rise to the veterinary institute in the world. The first veterinary institute at the Lyon, France, was to control the Rinderpest. Same the story in India. So beside these, Orthoparamyxoviridae is having a family called as the Haemagglutination genus. Haemagglutination genus. These are not species, the genus Haemagglutination. So Hendra and Nipah, these are the 2 important viruses, and initials has been taken to give rise to a new genus called Nipah. So Nipah virus is similar to that of this SARS-2, what the pandemic is occurring right now. It had happened in 80s, the Nipah. The Nipah virus from some of the fruitivorous bat transmitted to the pig, and from the pig it adapted to human. And every year we see the Nipah virus encephalitis occurring in Southeast Asian countries, including our country. Of course, that is not included in your syllabi, but for information, you must remember. These are some of the examples what we call as a species jumping or adaptation. From one species, the virus get adapted to another and causing serious disease manifestation. So Nipah virus is one such example under the Haemagglutination genus. Okay, this is in sort of classification. So the same things I'm just putting it in this tabular form. The highlighted one will be discussing on the Ortho-Ebola virus, the avian Ortho-Ebola virus 1. That day in the last set of this I discussed about the avian Paramyxoviridae 1, APMV. It was the earlier name. Now we call it avian Ortho-Ebola virus 1. So similarly, canine morbillivirus causes canine distemper disease. The Rinderpest morbillivirus that causes Rinderpest, and small ruminant morbillivirus, that's the causative organism that causes PPR, Pastidis petits ruminans. Okay, so these are different viruses under this Paramyxoviridae family. Okay, yes, we will get some touch to this family. We cannot finish up discussing all the members within a limited scope there in this particular class. So hope that whatever you learn, you will get a basic concept about the different classes and categories of viruses under DNA and RNA family, and the important members that you need to remember. Okay. Now let us come to the important properties of this group in general, important properties. So first thing is that they are some sort of an pleomorphic viruses. I mean, we cannot be having a definite shape. Some appears filamentous, some are again aggregated into a large spherical appearance. So basically, they are having the helical nucleocapsid symmetry. The nucleocapsids are arranging to form a cylindrical structures in helical fashion, and inside which the nucleic acid remain protected. So one such word called as the herringbone-shaped nucleocapsid is used. Probably if some of you have read the Hemingway's The Old Man and the Sea novel, then you'll find a descriptions of herringbone. This is like look like these are electron microscopic pictures of the this is the nucleocapsid structure, and this is the entire envelope. So you can see it doesn't have any definite shape. So as a close picture, it's revealed just like the spine of certain large sea fish, herring fish bone. The skeletons, how they look like. This is the appearance of the electron microscopes, and then this is very commonly this word is used, and we also very commonly use in the examination asking about what do you mean by this herringbone-shaped nucleocapsids, or which virus the nucleocapsid structure is herringbone-shaped. Please note it down. Now, coming to the nucleic acids, you'll find the size is not too big as compared to the previous what we discussed. There are 15 to 16 kilobases in size, moderate size genome, and they code for 7 to 8 different proteins, and that's all for the virus. With that, they are happy. So the genome is negative chains, single-stranded RNA, single molecules, non-segmented. That's why they're mononegavirales, negative chains. RNA virus. So they can nicely they can on the whole. Now we have their time already find their cells and share the transcriptions and transcriptions and they can synthesize own enzymes. Both grow in the cytoplasm. Okay. Here, interestingly, are space in intergenic spaces or spaces are there in between. They're individual genes. They doesn't have a operating frame. Rather, the genes are separated by conserved non-coding sequences in between. That's the characteristics of higher plants and animals. So there also we see this kind of nucleotide. So they replicate the cytoplasms and they produce inclusions when they grow inside the host cells. And a rough diagram about this virus, like through their cell surface glycoprotein spike, they are having an important glycoprotein called as the HN spike, Haemagglutination neuraminidase, through which they can land on the cell surface. More particularly, the sialic acid receptors they considered. And after that, the nucleic acid they simply fuse to the cytoplasms. They had another protein called as the fusion proteins that help in fusion of the envelope and the plasma membrane. Nucleic acid get pushed inside the cell, right? This is the method of fusion. And then, since they're having the RNA-dependent RNA polymerase utilizing that, they can go for transcription and translation. And altogether, you can see formations of structural and non-structural protein. Okay, so this proteins, the later part get aggregated, and again they comes out of the host cells through budding process. This is the budding process, and they carry this outer membrane of the host cell as envelope. This is the envelope. Okay, so just below the envelope, there is an aggregation of a protein. These are again virus-coated protein. This particular M protein, matrix protein, that gives us additional rigidity to the virus once they comes out, right? So we should have a general idea about this, how this virus replication takes place inside the host cell. Okay, so of course for individual families the details replication and transcription strategies will be difficult to remember, but hope that this will help you in giving a general idea. So this is the pictures earlier also I shared to describe how actually this replication stages that occur, the virus replication stages, the genomic replication I'm telling you. This is the example of the negative sense RNA viruses. So negative sense RNA viruses, they utilize the virion enzyme. That means the RNA-dependent RNA polymerase. Where from they will get this enzyme? They will take it in the previous replication cycle and it will be attached with the nucleic acids, come to the next cells, and utilizing that, this RNA-dependent RNA polymerase can synthesize the positive sense messenger RNA from the genomic RNA. So this enzyme, the virus must possess. It's not available inside the host cell. Then the proteins will be synthesized, and utilizing these proteins, as you know, and they are the early proteins. They again help to convert this negative sense to a positive sense RNA, and taking this positive sense RNA as a template, again the negative sense progeny RNA virus formation will be there. So thousands of copies of viral RNA will form at this stage, and then the assembly and the release will occur. Okay, this is the process they adopted by the viruses, their replication. So what you need to remember about the 6 major proteins and glycoproteins: the nucleoprotein NP, then the phosphoprotein P, matrix protein, then this underlined 2. These are 2 very, very important virulence attribute of this Newcastle disease virus, as well as the other members of Paramyxoviridae: fusion and Haemagglutination neuraminidase, HN. HN stands for Haemagglutination neuraminidase. Neuraminidase is an enzyme, and the important function of neuraminidase is a receptor destroying enzyme. Quite interesting. The virus utilizes this enzyme, neuraminidase, to comes out of the host cells, the release process. However, this Haemagglutination, they utilize it to land on the surface of the cell so that they can initiate the infection. This particular glycoprotein is named as Haemagglutination because it can cause agglutination of RBCs of certain species. So certain species RBC contain the receptor for Haemagglutination. It can cause agglutination, Haemagglutination. That's why they call us Haemagglutination. So these 2 are quite important for initiation of infection, and fusion is in proteins that help in the formation of fusion of cytoplasmic membrane of adjacent cells, leading to syncytia formation and cell death. So that's an important protein, this one. Then the L is the RNA-dependent RNA polymerase. As I said, here the genes are in between 2 genes, the non-coding sequences are present here, just like the eukaryotic system. Okay. So these are some of the general characteristics. And regarding the isolation and identification, that will be discussed along with the virus. So I feel this will help you, because already this NDV virus, the last Saturday you have attended the class. If any queries or anything will be there in between, you can ask the questions. During the class also, you can stop me and ask some questions, I can clarify it. Right? So this is like if we just discuss among ourselves, like which will be the most important disease in poultry, then obviously we'll say this NDB. So along with the influenza, of course, the influenza is not frequently occurring. Occasionally we see devastations by bird flu infections, but NDB is very, very common, and it's endemic in our country, and the birds frequently suffer and causes severe economic losses because of mortality. So in India, we call it Ranikhet disease because in 1926, first time in the island of Java, Indonesia, this scientist Doyle, he first identified the disease, could not establish the causative organisms, and then at the next year, 1927, severe outbreaks were recorded in a small island in England. It's Newcastle upon Tyne, so towards the Scotland area. That small island, there is a massive outbreak of the disease that occurred, similar to that of the Java outbreak in 1926. Exactly in the same year, 1927, the disease outbreak also occurred in a small village near Almora in Uttarakhand, near this Mukteshwar laboratory. You will get the Ranikhet is a small village where the disease outbreak was recorded. That's why in India, common name is the Ranikhet disease. Some common names, avian distemper, Korean foul plague, etc., are used, but like these words doesn't have much sense in the international forum. Nobody understand about the Ranikhet disease, so we have to speak about the etiology. So avian orthoepilvirus 1. Okay, so that's the causative organisms of Newcastle disease. So earlier it was called as avian paramyxovirus 1. Now it is renamed as avian orthoepilvirus 1. So if you want to clarify a doubt, then simply where you can go. If I ask you where from you can clarify this nomenclature, can anybody tell me? Yes, please respond. Hello. Please respond. Hello. Hello. Can anybody respond? Kisi Soden Bhutia, can you hear me? Hello.
Yes, sir.
Can you hear me? Sang Lamu. Can you hear me? Please respond.
Yes, sir.
Yes, sir.
What happened in between? You cannot hear me or what? When I ask some questions, why don't you respond? It's really it's a difficult situations. Virtual mode, we need to take the classes and online discussion. Well, do you have anything to say? Yeah, I ask simple questions like if you had any doubt about this name of the virus, where from you can clarify it? Yes.
Sir, NCBI.
NCPI will not be the appropriate place. It is the ICTV website, ICTV, International Committee for Taxonomy of Viruses, ICTV. NCBI is contain the GenBank, where you will get the genetic information of the viruses. Okay. You know the form of NCBI? Prepare plan. Okay. So ICTV websites, we can get and verify the latest viruses. And this morning only re-verified it. The Newcastle disease virus, they are classifying it's a mononegavirales. It's an order, Paramyxoviridae, it's a family, Haemagglutination is the subfamily, orthoepilvirus genus, and avian orthoepilvirus 1 is the species of Newcastle disease. Okay, so how this actually Haemagglutination and neuraminidase they appear and arrange in the virus is showing through this particular picture here. This particular green molecules, which is anchored to the envelope of the virus, this is HN. In case of this Newcastle disease, HN and they are fused together. Only one protein, say glycoprotein, they appear. Whereas in case of the viruses, you can get the H and N separated, right? And just near it is the fusion proteins, which is a virulence marker of the virus. Okay, so this Haemagglutination and neuraminidase are quite important in establishment of action as well as diagnostic point of view, where we can perform what's called as Haemagglutination. So the virus particle, if it is present in some clinical sample, we can mix up with the RBCs of the specific species, like the NDB virus can cause agglutination of chicken RBC. So we can mix it up. So what will happen? The virus will bind to the receptor on the surface of the RBC. As a result, the virus will interlink all the RBC together, and we can see some visible clumping. This is called agglutination, visible agglutination. The RBCs are quite big. When they clump together, we can see it by our eye. So this is called Haemagglutination, right? But as you know, this is not a true immunological test because there is no involvement of antibodies. So the more appropriate test will be the Haemagglutination inhibition, HI test. Okay, so these two tests are performed for diagnosis of hemaggglutinating viral disease. In HI test, Haemagglutination inhibition, what we do, we check whether the antibodies are there or not in the serum sample. Take the example like you vaccinate the bird and you want to know whether the bird is developing immunity. So you can collect the serum and you can mix up with a known virus and allow it for incubation for half an hour, and after that you mix up with RBC. So what will happen if the specific antibodies, they will cover up the virus. No more virus will be available to cause agglutination of RBCs. So as such, there will be no agglutination. This is called Haemagglutination inhibition. Okay. Whereas the virus and the RBC, if you mix it fairly, you can see the agglutination. This is called Haemagglutination. So these two tests, Haemagglutination and Haemagglutination inhibitions, are diagnostically important in disease diagnosis process. Okay, coming to the host, already it has been discussed. You know, the biggest problems of controlling this disease is a wide range. Almost 240 species of birds are known to harbor the virus. Some of them also suffer from infections, and sparrow, they may develop the disease. But many of the free-range birds, they simply harbor the virus in their intestine, a source of infection for commercial poultry. Okay, so the commercial poultry are highly this infection, and the infection is mainly through inhalation or aerosol infection. In contact, immediately they will transmit and disseminate the infection through droplets, and even by contaminated feed and paper, right? Till now, there is no vertical transmission established for the disease. Means through egg, it cannot transmit. As you know, in case of poultry, it's a very common mode of transmission. So many viruses which are transmitted through egg, embryonic eggs. Okay. Now, when you talk about the propagation of this virus in the laboratory for diagnosis purpose or vaccine productions, then you'll find both cell culture and embryonic egg systems are available for propagation. So, in embryonic egg, we inoculate through a lambda route of inoculation. I guess this picture is familiar to you. In our previous classes, we talked about how the different compartment inside the growing embryo, we can inject the virus material and the virus will grow in a living embryo, and we can see the changes and harvest the virus. So for Newcastle disease virus, we use this allantoic cavity route of inoculation, number 2, this particular route. So these are included in your practical classes. If possible, that sometime we'll have it and we'll done it. Okay. So if you just inoculate in amaranthine egg, depending on the virus, if it is a highly virulent, what we call as the velozenic, then the embryo will die within 40 to 60 hours of inoculations. Some are a moderate strain, we call the mesogenic strain, which can kill it between 60 to 90, and above 90 it's a lentogenic or least virulent virus. Likewise, these three pathotypes are described for MDV virus: velozenic, mesogenic, and lentogenic. Beside this, the cell culture, varieties of cell culture, we can utilize it. These are the examples showing it here, where the virus infections can causes syncytia formation, and then the cells will die gradually. If you can see this particular picture of syncytia formation, suppose these are the second embryo fibroblast cell culture, and these are the healthy cells, fibroblastic in nature, elongated cells. Can you see? So when the virus infection takes place, so the cytoplasmic membrane of many cells will clump together, and finally they will die. This is the syncytia formations, what we can see. All the nuclei will get aggregated together in the positions, and large empty spaces you can see in the monolayer. This is called syncytia formation, and that's a true indication about the virus group. I simply pick up from certain research articles where at different time interval how these changes in the cells are occurring. This is the normal healthy 24-hour infections, 48, 72, 96. You can see how the cells are destroying because of the virus replication. So we use the word cytopathic effect, CPE, to describe this kind of changes. This particular changes is called as a cytopathic effect, and more particularly, this cytopathic effect is syncytia formation. Okay. And this is basically assisted by the fusion proteins on the surface of the virus. So some of the pathotypes like viscerotropic velozenic, Doyle's form, neurotropic velozenic, Beach form, mesogenic, Budit's forms, lentogenic, Hisner's forms, and the last one is the asymptomatic enteric form, avirulent infection. So the different kind of the virus, avian virus 1 or avian Paramyxoviridae 1, the same virus. These different pathotypes were seen where this viscerotropic velozenic, neurotropic velozenics are highly virulent and are associated with the mortality which may reach up to 100%. So entire flock may die because of these infections. Okay, so basically it is a subepithelial infections where not exactly the epithelium, mucosal epithelium, the subepithelium, the lymphoid organs, the virus grow and multiply, and the inflammatory changes that occur. These are the cecum, and this is the cecal tonsil. I hope you understand the cecal tonsil. This is an important part where we can detect the virus at the postmortem. This is the proventriculus. This is another picture of proventriculus where it's showing the typical signs. Apical hemorrhage at the proventriculus. This is the gizzard. So this is some sort of an pathognomonic lesions for NDV virus. Always when we suspect, and then we cut open and see the proventriculus. But along the proventriculus, even the entire length of the intestines, we can see some diphtheritic plaques or membranes, and at the cecal tonsil. This is the junctions from where both the cecal arises. At this point, contain the cecal tonsils, where we can see the hemorrhagic lesions, which is diagnostically important. Well, there is a process of molecular method to pathotype it, and where on the basis of the fusion proteins, we can say whether it is a velozenic or it's a lentogenic strain. So at this stage, we'll not discuss much about it. Now, the conventional pathotypic system that are available for pathotyping of Newcastle disease virus, one is called as ICPI, intracerebral pathogenicity index, where intracerebral inoculation lead to the death of the chicks, and that is the score is recorded. And if the score is become 2, almost 2, then it's considered to be an velozenic strain. So gradually, when it's a mesogenic, it will come around 0.8 to 1, like that way, and the lactogenic, it is very less, 0.25, or even it may be 0. Similarly, there is another test called as IBPI, intravenous pathogenicity index. It's particularly performed in the 6-week-old, but these days we are not using the adult bird for pathotyping. And the last one is the mean death time in the embryo. We can inoculate the virus, and we can record at what time the embryo death occur, and say the velozenic 60 hour, mesogenic 60 to 90, lantogenic more than 90. So likewise, we can identify. I will just request you to record these 3 figures. These are the figures like the velozenic, mesogenic, and lantogenic strain. Lantogenics are least virulent strain, mesogenics are moderate, and velozenics are highly virulent. Okay, so in case of the embryonic egg, the velozenic always causes death within 40 to 60 or maximum 60 hours. Time will be needed, and the embryo will die. That's the property of velozenics. And mesogenic 60 to 90, and above 90, it's a lantogenic. Similarly, the ICPI index is also ranging. Say if it is 2, it's a velozenic, 0.8 to 1.48, it is mesogenic, and lantogenic is 0 to 0.25. Okay, if it occur. So these are the systems that people adapt to find out the particular virulency of this virus. So, like after entry of the virus inside the bird, it can circulate it through blood, and mostly the virus, they initiate infection in the subepithelial mucosal surfaces where the lymphoid aggregates are there, and severe inflammatory reactions are produced because of the stimulation of these lymphocytes, and that leads to severe inflammation produced in those surfaces, and that leads to a generalized conditions, and bird, they die because of massive damage that occur in those mucosal surfaces. So coming to diagnosis, clinical signs and symptoms no doubt can give us some information about the disease, including the postmortem lesions, but it need to be differentially diagnosed with diseases like the avian influenza or bird flu. So even like in case of IBD, sometimes and infectious laryngotracheitis, virus infection, some confusion sometimes it occur. So we need to go for laboratory test for detection and diagnosis. So HA test, Haemagglutination, and HI for antibody detections, we can perform it. Then very well we can isolate the virus in embryonic egg as well as in the cell culture, and subsequently we can find out the pathogenicity index. And other types like FAT is always there for detection of the virus in the cell culture. And since it's RNA virus, we can go for the RT-PCR test for accurate detection. So the virus-specific genes can be amplified and can be detected in a very short time. Now, when you talk about the prevention and control, for this particular virus, we are having some very excellent vaccine strain, and these vaccine strains are divided into 2 or 3 different categories of vaccines. One is that the lantogenic strains, very mild form of the virus, and generally the F strain, Lasota, which are very much prevalent in our market. They are used for primary vaccination, and usually within first week of hatching, after hatching, these vaccines are given. So once the bird, they took these primary vaccines with Lasota or F strain, any one of this lantogenic strain, then they are ready to take the mesogenic strains like the Komarov, R2B. These are available in our market, even the Mukteshwar also, but Mukteshwar is a little bit more hot strain than the other strains. So one of the problem with live mesogenic strain is that if the bird, they do not take the lantogenic strain, and straightway if you give the mesogenic strain, few number of birds may die after vaccination, because this is mesogenic strains, and they can cause some death in the bird also. These are all example of live virus vaccine. So always we should start with the lantogenic. Even the second also we can go for the lantogenic, and the third can be the mesogenic. So likewise, different area people adopt it, depend on the risk factor of giving the NDB in that area. Okay. So in the adult bird, we can go for inactivated vaccines, kill vaccines. We can use it, which can give protections in the chicks after hatching. Okay. So these are all live virus vaccines. So if you go for this kind of vaccinations, the vaccine virus may circulate in the populations, and of course, they may create some issues. That's why some countries, they prefer for recombinant vaccines, where they don't use this kind of live virus vaccines. So like recombinant fowlpox or recombinant Marek disease virus are used for giving protection into the bird. So in the Madras Veterinary College now, because the Tanu was, they developed one idaval vaccines. I mean, oral vaccines rather than idaval. I should say oral. It's like thermostable vaccines in the pelleted form, and they applied into this backyard poultry and getting very good results, and which will be very much pertaining to our conditions. So that kind of oral vaccines are also available for controlling MDB. Okay, so in the first week of life, the day-old chicks, or within 7 days, it's difficult to handle the bird. That's why the vaccination is given as aerosol spray. Some form, when they go for instillating in the nasal, 1 drop in the eye and 1 drop in the nasal passages to stimulate the bird. And mesogenics are usually as injections. We can give it in the wing. So likewise, we can taking care, we can control these infections by proper vaccinations and other zoosanitary measures, more particularly the entry of this wild bird to the commercial poultry. We must control it to get rid of this outbreak of Newcastle disease. So next important member under this Paramyxoviridae, the genus Morbillivirus, include a disease called canine distemper. Distempered, the temperament get lost in the animal. This basically this word are used to show some of the neurological symptoms that develop in the disease canine distemper. The nerve incoordination leads to some sort of an unnatural gait that produced by the animal, because of which is called as the canine distemper. And this is an quite important disease in the canine species, not only in the domestic, even in the wild canine families, even some Felidae also the disease has been recorded. So in India, the tiger and the lion, both this disease has been recorded, including so many other wildlife species. So it's really an important disease because the chronic forms may lead to the fatality, even the acute form. This is one such disease which infect almost all systems of the body. So talk about the hematopoietic systems, you can talk about the epitheliums and then nerves, etc., etc., respiratory tract. So almost all systems are affected by this particular virus. So common name is the heart pad disease because of the hyperkeratosis that occur into the foot pad. I think all of you already studied this infections in the pathology, and hopefully it will be a revision for you. But still try to get some more information if it is there. So this is a multi systemic viral disease, so gastrointestinal, central nervous system, respiratory, as well as the hematopoietic system are all affected because of this virus infection, and that leads to some conditions like leukopenia. And then when the virus they circulate into the blood circulation, then they appear a typical biphasic fever. So at a different time interval, the viruses come to the circulations, and the animal develop fever, intermittent fever. So in the respiratory mucosa, it produces the catarrhal inflammations, and exudations are occurred. Even sometimes the blood stained nasal ditches you may see in this kind of infection. However, in small animals like in pups, you can see severe diarrhea, hemorrhagic diarrhea it may produce because of the intestinal infections. And all these infections are basically the submucosal infection, not in the epithelium, it is in the subepithelium, because all of them they are the paramyxoviruses. They causes inflammatory reaction in the paraspecias and other lymphoid aggregates, submucosal lymphoid aggregates, and that causes inflammatory reactions in the mucosa. So Haemagglutination and frequent nervous manifestations are seen. If you go through the organisms, then you'll find this is that canine morbillivirus, the causative organism under Paramyxoviridae, morbillivirus genus, and canine morbillivirus is the causative organism. So almost the same kind of proteins that we discussed in the Paramyxo, Haemagglutination, neuraminidase, matrix, fusions, then polymerase, and then phosphoproteins are present. So one more information, so you can note it down. This particular virus, canine distemper virus, they share some similar antigenic structures with measles virus of human and Rinderpest and PPR. So sometimes we call it a viral triad, CDV, measles, Rinderpest. So they have their cross protections, like that against canine distemper virus can react with the measles and the Rinderpest virus. Even up to the extent that people say, if you have the measles vaccines, then if you give it to the dog will be protected to canine distemper virus. Okay, so they're cross-protecting each other. Now, this canine distemper virus can be propagated. All these three systems available for virus isolation: cell culture, embryonic, and the laboratory animal. So obviously the cell culture will be most preferred, but at the same time, embryonic eggs also support the growth of canine distemper virus, but this is least preferred because we have to go for 9 passes, only then the virus will start adapting to the eggs. So it's take a lot of time in that process. So cell culture is the best method where we can use some canine cells like Medite derby canine kidney cells, where we can grow the virus, and it produces typical syncytia. Okay, so this virus, as I said, they produces inclusons. So they produces intracytoplasmic as well as nuclear inclusion bodies. Okay. At this point, you may ask me questions like the virus is totally replicating in the cytoplasm. Then why it is producing intranuclear antibodies? So this is an Paramyxovirus is always replicate. They have nothing to take with the nuclear region, but why the inclusions are seen in the nucleus? So this question could not be answered for long period, but now it has been explored that in the canine distemper virus infections, in the presence of the viral proteins causes some hybrid destructions in the nuclear area, and this is simply deposits of degenerated host cell microfibers inside the nucleus. It's not the viral aggregate, but that degenerated cellular structures appear as an inclusions in the nucleus. So many attempts to detect the presence of viral capsids in the intranuclear inclusions was failed, and that reveals that that is not a viral structures, it is some cellular debris only. Okay, so in this canine distemper, both intracytoplasmic and internuclear inclusions are same. So these things are diagnostically important. This is a typical syncytia formations so that we can see the cell nuclei get aggregated and empty spaces are produced in the monolayer. And this is an intact monolayer. You can see when the virus infection takes place, this kind of syncytia and destructions takes place. Okay. On the other side, this is the green things are the FAT test, fluorescent test, where using this fluorescent isothiocyanate, the level antibodies to stain the monolayer cell culture infected with the canine distemper virus. So the locations where the viruses are present are producing apple green fluorescence under fluorescent microscope. So likewise, the presence of the viruses can be detected directly into their the appropriate locations, what we call in situ detection of the virus. So this effort is really important in viral disease diagnosis. So if you go through it, then you'll find not only the domestic dog, even the wildlife. Some slides like this is in BBC News. This says Asian tigers are at risk from a domestic dog distemper virus. So it's really happened, and some confirmed reports are there. Even like what is killing lions, and it is detected that the Paramyxoviridae infections, lion is the cause, and distemper occurred. So not only this domestic, even the wildlife are also susceptible to this infection. So mainly by in contact, aerosol transmissions and direct contact is the main route. Even in the chronic case, the viruses are excreted in the urine, and in contact may cause dissemination of the disease. So by secretion and excretion, the viruses is coming out. Good thing is that the virus is not very much stable in the environment. It's hardly for say a few minutes it can survive in dry condition. If it is totally dry condition, the virus readily inactivates. And then, but close contact may lead to transmission of the disease, the virus to new animal. So once it enters to the mucous membrane, then the macrophages, they become the second transport within the body. So they carry the lymph nodes, and then in the lymph node, the virus will start infecting the lymphocytes, and they can establish infection very well in those cell population. And then they are disseminating to the different parts of the body, like in the Paraspetses, as I say, all the submucosal lymphoid aggregates, the lymphocytes, they carry the virus to those areas, and then they start producing inflammatory reaction there because of the cellular damage and immune stimulations. You can see the inflammation, right? So see, by 5 to 7 days the incubation period. After that, yeah, one more slides we can see. Sorry, oh, it's not here. Okay. So the virus, they first initially they affect this because epithelium in the respiratory tract and the gastrointestinal tract and exhibiting severe diarrhea and the respiratory distresses. And gradually, when the immune systems will be stimulated, they will persist it for the longer period. And we see once the acute phase of disease is over, then chronic type of infections persist in the animal, and they are gradually taken to the central nervous systems where the encephalomyelitis conditions develop. And because of that, the nerve cells, they lost their normal functions of controlling the nerve impulses, and the animal exhibit distemper-like symptoms like myoclonus, twisting of muscles, and then there will be some jerky movement and the respiratory distress the animals they exhibit once this stage is over. So not only that, even what we could see in the growing pups, where the dental development is not complete, then it can causes some improper teeth development or dental caries are produces in the pups when these infections that occur. Besides this keratoconjunctivitis, we can see and optic neuritis. So varieties of systems are affected. So there is a condition called as the old dog encephalitis, where the animal they exhibit chewing gums appearance as if the animal is chewing some chocolates or something like that. These are basically the incoordination, nerve incoordination produced in the animal. So deterioration of mental abilities. So these are all reflections of the neural signs and symptoms. So enamel hypoplasia, this is what I said, and the pups we could see, and then the lesions are produced. So this is one hyperkeratosis, even nasal dentures, respiratory, even blood-stained mucosa comes out, and foot pad, that's why it's called as the heart pad. Yeah, these are some of the things that I could collect it. Coming to the diagnosis, obviously the clinical signs and symptoms gives us a good information. However, it is again confusing with some other disease because all the systems are affected, starting from nervine symptom, gastrointestinal tract, respiratory tract. So we need to go for confirmatory diagnosis where certain tests like the CSF fluid, we can detect the virus-specific IgM or IgG to find out the acute or the chronic infection stage. And obviously, we can go for the cell culture and membrane-derived system to isolate the virus and for the confirmations by the laboratory test like AGPT, antigen capsular ELISA, and FAT test we can perform it. So this diagnostic approach is similar that with other viral infections. So to say, yeah, this particular slides I just want to focus. It's an excellent slide on FAT. So we can collect the blood and the Buffy coat. We can prepare a smear. Lower one, this is the Buffy coat smear, where the fluorescent antibodies test is performed, and these are the infected lymphocytes and where the apple green fluorescence are producing. Okay, so this is another where the cell culture, the FAT is performed. And they're using one nuclear stain to stain the nucleus of the infected cells, so to get a contrast or apple green fluorescent red. This way, this apple green fluorescent indicate the FITC stained virus aggregates. So FAT is an important test because directly we can detect the presence of the virus in the cells, which is always considered to be a gold standard, although we cannot see it by our normal light microscope, but by FAT test using a fluorescent microscope, we can detect the virus. Okay. So, like the other viral infections, we had good vaccines for controlling the infection, and then yeah, only a few number of you in class. I don't know why. So one particular strain, please note it down, the rock-borne strain, attenuated strain, rock-borne strain. This is quite commonly used in India as an live attenuated strain to vaccinate the animal. And most of the time, in case of the dog, we are having the multi-component vaccine, several pathogens together or put together in one vaccine vials. Sometimes we call it a multivalent vaccine, including multiple pathogen. If you can remember in our discussions with the leptospirosis, I said about a vaccines called Megaveg or Pentadog. where the bacterial and the viral agents are clubbed together to have this vaccine. So this is also a part of that particular vaccine. So usually we have to be very careful about this maternal antibody. So what happened, mothers, if they are vaccinated or are naturally infected, they pass the antibodies to the pups and through colostrum, and they circulate in the blood. So if the pups have the maternal antibody, then we should not go for vaccination. Usually they decline after 6 to 8 weeks. Then this is the time, like after say 1 month only, we should start vaccinating the animal with this. And then after 28 days, again, we should go for the booster vaccination. And so once we give the booster vaccination, then annual revaccination is suggested. Please remember, in case of the canine, most of the canine vaccines, after giving the first shot, which is generally given after usually 1 to 2 months, because the maternal antibody persists that period. And if you vaccinate the animals when it is having the maternal antibody against those pathogens, then these antibodies will neutralize the vaccine virus. As a result, the immune stimulation will not be proper. So that is why we have to wait till the maternal antibodies diminishes from the blood circulation. Usually after 1 month it happens. Then we should start vaccines, and after the first vaccination, after 1 month of the first vaccination, we must give the booster dose so that the animals will remain protected for 1 year. So by maintaining this kind of proper vaccination procedure, we can get rid of the infections. Okay. As I said that earlier, it happened like the measles vaccines, people use it to vaccinate the animals. But nowadays, the vaccines are quite commonly available in the market, and that such type of cross-protective vaccines doesn't need it. So again, the other preventive measure, as I said, that the virus is not stable in the environment. Hardly for a few minutes it can survive outside the host. So disinfecting the premises will readily inactivate the virus, and always the close contact is needed. So to avoid the close contact, we can get rid of the infections. Okay. Yes. Do you have any specific question to be discuss? Can you hear me?
Yes, sir.
Okay. So at this point, I just want to talk with you. You can switch on your videos if you wish. CR is here. Class representative. See, last week, we had a common class with the Jaluki Veterinary College and Professor Rajakumar. He took the class on the Saturday, and we thought of conducting the same class tomorrow. Are you going to attend the class, or should we continue after your exam? Accordingly, I need to. Yes.
After exam.
Now immediately you respond when question you do not respond. Sarup.
Yes, sir.
Can I see you? Can switch on your video. Hello.
Yes, sir.
Sarup.
Yes, sir. Yes, sir. There's some network problem. I'm trying. Sir.
Okay. I understand. Okay, I understand. After exam, you mean? No? So I need to convey the message to them, and then only possible. Is it same things everyone feels?
Yes.
So yeah, please request your CR to pass message to me. Hi. Everything is all right? Okay, please ask your CR to pass a message to me. Gargi Sakuntalam, she has left the meeting. Okay.
No, sir, I'm here.
Oh, okay. Yeah. How are you?
Sir, I'm fine.
Situation is improving in Delhi.
Yes, sir, it's improving.
Take care. Have you able to take your vaccine?
Yes, sir, first dose.
Okay, that's great. That's great. So second also try to get. Which one you have taken? Covishield or Covaxin?
Sir, Covaxin.
No, Covaxins. Okay, inactivated vaccine. Okay. Here in Mizoram, we are getting the Covishield only. So take care. In the class when we say that hello, somebody should respond, I feel you should respond. Please try to have some interactions, and then we can discuss. As the Swarup says that tomorrow it will not be much convenient, I don't know, but still come on representation from your class representative. If he proceed to me, then it will be easier for me to discuss with the Jaloki professor. Okay, should I continue or right? Shortly, a few slides are there. Next, another important infections under the Paramyxoviridae family is under again the morbillivirus genus is the Rinderpest. As you know, this has been now eradicated globally. So it's called as the cattle plague because of the large-scale death it cause and severe losses encountered. And in fact, when the first outbreak occurred in African continent, it was some sort of famine that occurred because of this Rinderpest. And it has been seen in almost many species of Artiodactyla. Okay, and this is mainly characterized by severe hemorrhagic inflammations in the gastrointestinal tract, leading to severe diarrhea, as well as the blood cell, the leukocytosis and occur. These are some of the pictures taken, and the virus, of course, very shortly we'll discuss. It's not even included in your syllabus, but I feel that one should read about the Rinderpest, should know it. This is such an important disease. Earlier it was there. So the morbillivirus and the same proteins that you could see with some non-structural protein C. This is another highlight. So it was some sort of the pictures. This one I'm more interested. In 1980s, you can see this area where the disease was prevalent, and out of extensive vaccination and segregations, gradually it is come down, and by 2007 last it was detected, and then Ethiopia probably in this African continent, and after that no more infections could be detected globally. And by using the effective vaccines grown in the primary cell culture, as well as with some cell lines, the Vero cell adapted vaccines that become the most crucial in eradicating this particular disease. Okay, so the disease clinically is having 3 phases: prodromal, mucosal, and diarrheal phase. This mucosal means the inflammation of the subepitheliums, and the entire gastrointestinal tract leads to severe dehydration, and the death occur in the animal. So there is one strain called a Saudi 181 strain. You can note it down. This is one such virulent strain which can cause 100% mortality in the Rinderpest infection. So this was in the Saudi Arabia. It was characterized and causing severe damages, and mostly the pathognomonic lesions. You can see the tiger strip or zebra strips that produce in the cecum, colon, and rectum. So this is something the mucosal enfoldings, like in the epithelium, the inflammatory reactions are produced, and it appears as an red hemorrhagic strips and the white, red and white, like that way. When you fix it in the formalin, they appear as the dark and white, similar to that of the skin coat of zebra. That's why it's called as the zebra stripping or tiger stripping. Okay, so these are the intestinal diseases that produce in this disease, and almost all methods of virus isolations and common tests like AZPT, counter-immune electrophoresis, FAT, these are can be performed and extensively performed in our country when the disease outbreaks were there, and the diagnosis were done that way. Okay. Now coming to another Rinderpest-like disease. Now this is very much prevalent in our country, and this is one of the major cause of goat mortality, viral disease of poultry. Goat. It's called as the PPR, Pastitis petits ruminants. The meaning is the small ruminant infections. So it is in morbillivirus infections. It's having several names in India, particularly in Andhra Pradesh and southern India. We call it a kata. Kata is a name for catarrhal inflammations and goat plague, large-scale death, pseudo Rinderpest-like disease, because the other clinical manifestations are similar to that of the Rinderpest. So likewise, several synonyms are there of this particular infection. So this is basically a systemic infections of goat and sheep, and will be characterized by all these symptoms of fever, occlusal ditches, erosive stomatitis, enteritis, pneumonia, and death. So here, both respiratory as well as the gastrointestinal tract, the infections are seen, and mainly by the blood cells, they disseminate the infections, the macrophages as well as the lymphocytes. So the suffering goats, they exhibit this kind of catarrhal inflammations. The catarrhs are produced. So that's why it's called as kata in southern peninsula in India. So nasal ditches indicating the severe respiratory problems, but at the same time, severe diarrhea is another consequences. Here you can see the OIE map where the prevalence are showing it here. The color intensity indicates the intensity of the disease, the number of outbreaks and the severity. I mean, our country, it is highly endemic. What is PPR? So invariably, we'll encounter this condition in the field and should know it very properly about this morbillivirus infection. So in 1987, first time it was detected because of animal migration has come to our country, and then it is spread to all corner of India. So basically, the disease is transmitted by contact with the sick animals. So the viruses mainly remain in the affected animals, and they excrete it, and close contact lead to the disease dissemination. So as I said, some of the outbreaks, the mortality may reach up to 100%. So obviously, it will be an high-ranked viral infections of goat. So goats are more susceptible than the sheep, whereas the blue tongue, we see that are mainly affected. So coming to the etiology, you'll find it's the Paramyxoviridae subfamily. Paramyxoviridae is the family. Genus is morbillivirus, and the causative organism is small ruminant morbillivirus. Earlier it was called as Pastitis petits ruminans. Now it has been renamed as small ruminant morbillivirus. So virus can be grown in the BHK and the Vero cells that produces syncytia and total destruction of the cell. However, in certain specific cell line like lymphoblastoid marmoset cell line, they produce some clock face appearance. This is one clock face appearance where the virus they causes aggregation of the cells, look like the clock face, and this is a specific cell line used for growing this PPR virus. However, very well we can grow in Vero as well as the BSK21 cell line, baby hamster kidney 21, where they produce severe damages. Now, coming to the pathogenesis, the viruses, they enter through the nasal passages, and once it enters through the nasal mucosa, they initially grow in the lymph nodes, and then through the lymphatic streams, they are disseminating to the regional lymph nodes, and then the respiratory and the alimentary tracts, the secondary viremia phase, they infect. So severe inflammatory reactions are produced in the respiratory mucus as well as in the alimentary mucus, and that causes the damage in those areas. And the alimentary tract and the respiratory tract, so alimentary tract infections lead to severe diarrhea and dehydrations exhibited by the animal and subsequent death. And in the respiratory passages leads to consolidation of the lungs and severe pneumonia are produced. Okay, this is another diagram. I could get it from internet, where it's showing like the virus entered, and then through the dendritic cells, macrophage, and the monocytes, they carry it to the lungs, and infections occur. Then the secondary viremia, the other lymphoid organs are affected, and that leads to severe inflammatory condition in the intestine as well as the respiratory mucosa. So that's causing to sometimes 100% mortality into the animal. So obviously all the signs of respiratory signs and the diarrhea, I can see it. The damages or the inflammations in the intestine is so severe that leads to blood stain diarrhea, and then the same zebra strips can be appeared. This is another pictures that we could see in the PPR infection. And here the lung consolidations. So to such extent the lungs are damaged, then obviously it will cause death in the animal. So coming to the diagnosis based on the clinical signs and symptoms and postmortem examination, which are very typical about the PPR infection. However, we can go for the laboratory test and isolation procedure as well as the molecular detection methods for specific and rapid detection of the virus. So, this kind of some lateral flow test, the rapid antigen test kits are also available in our country in the market for rapid detection. So these are pain site tests. Within that, from premises, we can detect the disease. So, for controlling the infections in our country, we are using our vaccine strain. It's a Sungri 96 strain. It's the name of a place where the virulent virus was isolated, and later on, this virus was adapted to the Vero cells, and this is a live attenuated vaccine strain, so which can give very good protections into goat. One should go for the vaccinations, and that will really protect the animals for occurrence of PPR. So definitely, just like the Rinderpest, the PPR also one day will be able to overcome and can eradicate the disease. Many countries in the developed countries, they already eradicated, and there is no crash of this kind of disease in Australia, New Zealand, and all European country, and both North and South America. You will not get this kind of disease. So very well by effective vaccination. This is the task of the veterinarians, like how they can go for controlling the infections by proper vaccination. So I request all of you to note down this Sungri 96 strain, which is prepared by the Indian Immunologicals and are available in our market. So it is branded as the Raksa PPR, Indian Immunological Company, they prepare it. So this is a Verocell adapted live attenuated strain of PPR. Okay, thank you very much. This was I prepared it for today's presentation, and then after the examinations, we'll again continue. We have to cover up all this RNA and the DNA virus family in our discussion. Okay. Thank you very much. If you have any specific queries or questions, you can ask or you can text me so that in the next class we can discuss it. Today attendance is very short, only 26. Yeah. Thank you very much then. I'm closing the link.